Welcome. My name is Kelly Truax, and I will be presenting our research under the MTV Consortium focusing on quantifying moss response to contaminant exposure using laser-induced fluorescence. More specifically, the research is placed within Thrust Area 2 and is focused on developing technology and methodology for non-proliferation by detecting contaminant deposition using and observing biota. Our primary goal is to develop a technique that can detect contamination of metals and radionuclides of interest from a variety of sources, such as nuclear fallout, mining waste, nuclear waste, etc. The project utilizes the BioFinder, which is a remote laser-induced fluorescence detection method that was originally developed by another team at our University of Hawaii under a grant from NASA to identify organic material. In our project, we use this unique design of the BioFinder to observe changes in biological material like chlorophyll composition in plants. Our study pairs this technology with observations of moss to detect any change that may occur due to metal accumulation in the moss. Mosses are unique in that they've long been used in environmental monitoring, they are easy to work with, and they are a robust and resilient species. However, traditional approaches to environmental monitoring involve sampling and laboratory analysis, which can make the methods labor-intensive, destructive, and often expensive when used on a large scale. Due to these constraints, a remote sensing approach could prove to be a viable alternative. For the current study, we tested the hypothesis that metal deposition would induce a physiological response in the moss samples, that response could then be detected using laser-induced fluorescence, which we'll refer to as LIF, and images of that fluorescence could be analyzed for changes to detect contamination. With the NNSA mission firmly focused on the detection and prevention of militarization of nuclear weapons, our work aims to develop a non-invasive remote technique to detect biological response in vegetation after exposure to a metal of interest, specifically from mining or nuclear waste in the production process. Our end goal is to develop a remote sensing technology that could survey areas of interest to identify the presence or absence of contamination, pinpoint its source, and aid in bioremediation. The methodology was divided into two parts, with the first focusing on the cultivation and treatment of moss, which was implemented after receiving input from Dr. Wendy Kune and Dr. Laura Tova at Savannah River National Laboratory. First, we cultivated moss in a grow tent, seen in the images on the right. One moss mat was left as the control and only given deionized water, while the other 3 moss mats were treated with various concentrations of copper chloride. Previous studies have documented that copper is readily taken up by and induces a known reaction in mosses that could directly affect a change in chlorophyll A-B ratios. Said changes could then be identified by comparing treated and untreated moss samples using LIF. The experiment was conducted for 10 days, with each moss receiving a copper dose every 48 hours, or 5 doses in total. The table here shows the total cumulative copper concentration in micromoles per centimeter squared given each moss mat over the course of the experiment. Actual moss copper uptake was monitored by analyzing copper content of 3 fronds from each moss mat each day. Part 2 of the methodology involved imaging the moss mats using the BioFinder and then using image analysis to identify changes in the images as a result of increasing copper doses. Each moss sample, treated and untreated, were imaged under LIF every 0, 6, 12, 24, 36, and 48 hours. Then the process would repeat each time a new dose was administered. Imaging was conducted under daylight conditions, or under fluorescent lights in a room, at a half-meter distance using a CMOS camera. Images were collected under a 532 nanometer green laser and a 355 nanometer UV laser individually and in tandem. These images were then processed in MATLAB to extract the red, green, and blue decimal code values for every pixel. These pixel counts were then compiled into density histograms seen on the rightmost image. These RGB density histograms were then used to define the differences in images between the control and each of the treated trials. To do this, we used 2 different methods to compare the histogram curve. On the top row, histogram of an image taken before treatment, what we often call a day 0 histogram, or control for the trial, is shown on the left. A histogram of the image taken after the completion of the full treatment experiment, or 10-day histogram, is on the right. Both images are the same moss sample used throughout the experiment as trial 4, the one with the highest contamination level in the experiment. We compare the two curves by plotting them together and finding the area that does not overlap between them, meaning the difference between the curves, as seen on the bottom left. The second method is called dynamic time warping, or DTW for short, and is a curve-fitting method that has been employed in image analysis of handwriting. It finds the closest distance between all points between the curves and sums that difference. We first applied these steps to the first 48 hours of the experiment to demonstrate that 48 hours is a sufficient time to wait for the moss to have a full response to an individual copper dose. On the left, we can see that there is a larger difference between trials initially, soon after treatment followed by a slight decline after 36 hours. We plot this on the right based on dosage to show that within 5 minutes of dosing, the imaging technique can detect a measurable response with all trials clearing the control's mean plus or minus 2 sigma deviation, or a 95% confidence interval. Of importance is also that on trial 1 control images, we don't see much change after wetting between day and night, or in general over the course of the experiment. This allows us to rule out any environmental variability as a cause for change in the images of the treated trials. We then went on to analyze all 10 days of the experiment, creating both 1-color and 2-color analysis of density histograms from the images. In 1-color analysis, just looking, for example, at the red channel, we can see an increase in copper dose results in a shift to darker, lower RGB decimal codes, which appears as more peaked and more right-skewed curves. 2-color analysis compares red and green color channels to observe how their relationship changes with increasing copper. Increased copper leads to longer, narrower loops. It is clear to see, at least qualitatively, that all trials by day 10 are distinct from the control. We can apply the density difference and DTW methods described earlier to quantitatively express differences between images with increasing copper added to the moss map. The histogram difference for all colored channels increases with increasing copper dose. These are derived by comparing images taken at each day to the trial's day 0 control image. All 3 colors show a similar response here. DTW results are very similar, showing clear departures from day 0 images with increasing differences each day. The leftmost figure here shows density difference. The middle plot shows DTW applied only to the red channel. The two methods are in good agreement with the distribution of points, but there appears to be a slightly better separation from the trial 1 control when looking at the use of the DTW method in the center. Both methods clear the mean of the control plus or minus 3 sigma, or a 99% confidence interval, and it appears that multiple small doses, or even a single large dose, results in the same difference. Because DTW is in good agreement with the density difference method, it was deemed that DTW could be used on the more complex looped red versus green shapes where density difference was not applicable. The result is the figure all the way to the right, which not only shows better separation of treated moss images from the control, but also indicates better separation between individual images from the same dosing period. This perceived greater sensitivity makes 2-color DTW potentially more viable than either 1-color analysis due to less overlapping between data points. This was a proof of concept study that utilized only copper, but the results showed great promise for a laser-based methodology for field application. Continued work aims towards real-time detection of metal contamination and its distribution, which would help eliminate the need for extensive sampling and aid in targeted sampling and analysis. The advantages of this method are that it can help in the identification of presence or absence of contaminants, as well as their source and spatial distribution. The technology is not limited by time of day and has flexibility to be used at distance, unlike near-infrared, which is currently prevalently used in agriculture. The biofinder is also low energy and in a portable state, though further development is needed for onboard programming and housing. Drone deployment of the remote sensing technique would be the ultimate end goal. Thanks to MTV's support, 1 graduate and 2 undergraduate students are helping with research efforts. And we've been able to collaborate with national laboratories to develop experimental designs, discuss future collaborations, and whether those be site visits or internships. MTV has also had an impact on connecting our students with workshops like the Next Gen AI for Proliferation Detection and lecture series like the NSSC Alumni Speaker Series. Without MTV, potential field application of the technology at Savannah River National Laboratory may not have been possible. We also have had the great opportunity to discuss collaborations and internship opportunities with Pacific Northwest National Laboratory and Lawrence Livermore National Laboratory. In conclusion, the study has proven that LIF can be a viable remote sensing method for detecting metal exposure in moss and potentially other plants. That we can detect physiological change, that single color or two color analysis shows separation from the control to a 99% confidence interval, gives us great hope for the future. Though this was mainly a proof of concept study and our lowest dose that we tested, though easily detectable, was only sensitive enough for extremely contaminated sites like the Morgul copper mine in Turkey. Thus, further work on sensitivity is of utmost importance. We do have preliminary data on zinc and lead that shows promise, and we feel confident to begin preparation for uranium testing as well. We will work with the national labs to obtain the desired detection limits and sensitivities that they desire and continue to develop real-time image processing for field application. Finally, we've already begun to take our next step. Several changes in the biofinder have led us to have improved detection in the 1 to 10 nanomolar per centimeter squared range that we will need for our future research. This needs to be refined, and a relationship between sensitivity and laser distance from the sample needs to be established. In the future, we plan to do full experiments not only with more metals, but mixtures of metal. We plan to begin application to other vegetation, conduct chlorophyll tests to see how chlorophyll A and B ratios affect our images, and also to do various stress tests like drought, temperature extremes, and various photo periods to compare environmentally stressed samples to metal contamination stress samples. As mentioned before, we have 2 undergraduate students who will be working on cultivating new plants, on image and chlorophyll analysis, and to develop their own project. We're very interested in continuing to foster collaborations with the national labs, and we will be working to further our understanding of data analysis and machine learning to help assist with real-time image analysis and contaminant identification. With that, I would like to say thank you once more to the MTV Consortium for supporting our continued research and the opportunities that affords us. And thank you to you for listening to this presentation.